Hello everyone.
Hello.
Any doubt?
Hello.
Hello Paul. How are you guys?
Good,
Are
and
you
you?
doing okay? Perfect. Rakhi, what about you? Any doubt?
No,
No, ma'am.
No. Okay. Are you enjoying it? All of you?
I'm not enjoying it. I am breathing it.
Okay. Okay. Okay then. So in today's session, we are going to see the enrichment. So in the enrichment, the two topics will be covered. That is the functional enrichment and the pathway enrichment. Okay. So first we will look into the pathway enrichment. So what is the pathway enrichment? Can you tell me?
Can I give it a try?
Yeah, Paul.
I think I'm just guessing, so I'm not very sure. But I think the pathway enrichment
Uh-huh.
um, allow to find I will say the protein that interact, the different
Okay.
partner that interact with that protein.
Yeah.
Yeah.
So in the pathway enrichment analysis, what happened? Like we are going to see in which pathway our gene is involved or gene is highly enriched. So that we are going to find out here. So to find out this information, we are going to use one of the database, that is KEG Mapper, color and search pathway. So in this KEG mapper, we are highlighting the genes from our list with the different colors. Why we are highlighting the genes with different colors? Any guess? Tell me.
To check maybe upregulation or downregulation. Or
Yes, sir. Yes, sir. So in this KEG database, already there are a lot of genes that is present here. Okay. And those genes are highlighted with the green color. Okay. But in order to differentiate our genes, that is, our genes are highlighted with the red color here, and here it is green color, right? So we want to see only our genes, right, in the pathway. So that is why we are going to highlight those genes with the different colors. So let's see how to create this pathway information from the KEG database. So you can see here from the UniProt, we retrieve this data, right? And along with this L column, you can see there is a HSA ID. Can you see this one?
yeah,
So, what we have to do, just copy this HSA ID of your from the upregulated genes and paste in the new sheet. Then this is the ID for the upregulated, right? So we are going to highlight those genes with the red color. So I will write here red and then autofill it. For the down regulated also same thing you have to do, but you can see the down regulated genes here, it is in the green color, right? So I already told you whatever the gene that is present in the KEG database that are already in the green color, so we cannot differentiate the down regulated genes, right? So what I will do, I will just give this color blue to my down regulated genes, then autofill it. Okay, now Are you doing along with me or you will practice later on? Tell me.
I will
I will
practice later.
practice it.
Later? Okay, perfect. Now, if I try to just copy this entire data and paste those HSA ID here, and you can see this is the HSA code here, right? So this data has been taken from human organism. So I will just select the HSA from here. But if your data it is from the other species, that time you can generate the code also. Get the organism code here, and you can just type here, for example, if my data it is from cow, just write here cow and you will get a code. You can see here BTA. So the BTA code is for the cow. Okay.
Also, what is the usage of this code?
Which one, sir? Then
That,
only
yeah.
it is going to take, sir. You can
Mhm.
see if I try to just select this reference and execute this,
Okay.
it is not going to show any pathway. Okay.
Mhm.
So you have to put the data first here in this box and you have to generate the code. Okay. Then only it is going to take those IDs. Okay.
Okay.
Then just select the HSC code from here and execute it. Again, you can see there is no pathway, right? Why? Because in this ID, you can see after the code here, there is a semicolon. Can you see that?
Yeah.
So it is not going to take those HSA ID who is having semicolon. So what we are going to do, just copy the entire this HSA ID and paste here. And we have to remove this. Go to the data and there's the option text to column, delimited, next, go to other and put here. Then finish. Okay. So it has removed. So what we can do now?
Replace the
Yeah.
first column.
So you can replace. So now if I try to just entirely copy this, clear this, then HSA code here, then execute. Now you can see total 9 pathways are involved. And in this sheet, you can see along with the HSA code they are giving you. This is the HSA code for the metabolic pathway. And the species name you can see here, human. And in the brackets, these are the number of genes, okay, that is involved in this particular pathway. Clear till here?
Yeah.
Now, for example, if I want to work on this tyrosine metabolism, what you can do, just open the HSA code from here. Now, you can see our gene, it is highlighted with the red color here. You can see the gene here, FHA D1. Okay. Now, this is the pathway it has created. And also, if you want to retrieve more information regarding this pathway, you can go and select the pathway entry. Now, again, the entry name they are giving you, the pathway name, and the pathway figure. So, whatever the gene that is present here in this pathway, you can see below. So, this is important, the genes that are involved. Again, if you want to go like you want to study more about this particular gene, so you can just go with this ID or with the EC number or the KO IDs, you can get that those information. Clear? Any doubt till here? So this is how we can highlight our genes in the KEGG database. Oh, there are 3 genes here. Okay. Now, every time I cannot load this HSA code in the KEG database, right? So what I will do, I will just simply copy this entire data and paste here. So I will write here pathway enrichment. Okay. So now there is a space right in between. So just select the A column, then go to data. There is option sort. Column A, make it okay. Okay. Now in this, you can see it is common human. So you already know the data has been taken from human only, right? So next thing is what we are going to do. Just simply copy this till here. Then just select the A column, then type Control H. So we are going to replace this, okay? Clear? Or you can replace with this also if you want. Control
What are we
H.
replacing? I don't understand why.
This one, Paul. This is common, right? Human.
Yes. Yeah.
Already we know this data has been taken from human only. So what I will do, I am going to replace with the hashtag, for example, now. Okay.
Okay.
Then
You are
replace it.
removing, yeah?
Yes, yes, yes.
Okay,
We are
okay.
replacing with the hashtag. Can you see
Yeah,
that,
okay.
Paul?
Yeah, I see.
Okay, because already I know this data has been taken from human. So two times it is telling you human, Homo sapien. Can you see that? So I am just replacing with this hashtag. Can you see that now? Okay, because in the research paper, I am going to tell you how to load this data. Okay, so this type of data we are not loading, that you are looking at this paper. Can you see that? So this is how we are not reporting this. So I will tell you how to report that. So you can see here again, just select the A column here, go to data, there is option text to column, and then delimited. Next, in the other, put hashtag here, then finish. So what I will do, I will just put insert one row above, and I will write here name of the pathway and number of genes involved.
One question.
Yeah. Paul?
Are we not missing one pathway? Because I know that the first one has three protein that were included. Here we are
Which
having just one, the number of gene involved. I think that
this one?
they were a pathway. Ah, okay, okay. It is at the yes fifth position. Yes, position 5. Yeah, it's fine. I'm
Okay.
seeing now. Yeah.
Hmm. Then you can just type a control H, then put the bracket this and replace all. Okay. Now, for example, if you want to download this, for example, you are working on this tyrosine metabolism pathway, you can download this figure also. in the PNG format. Then mm, you can insert it in this Excel sheet also. There is an option, insert option up there. Go to picture. Mm.
Is there any way to associate that picture to the gene so that we can just have a kind of link
Which
from the
link?
when I link, I say a link so that if you click on the Excel sheet, it
Uh-huh.
can also open the file on the Excel sheet, maybe just aside, just beside. I mean,
Um,
if you click on the HSA ID, since you want to
where?
associate each gene with
Uh,
each pathway with the images,
mm-hmm.
I want to know if you can do it with Excel so that if you click from the Excel, immediately the a part where you can have the pictures that
No,
you
no,
have
no,
downloaded.
no, 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 no. That we cannot do.
Okay.
We need to insert that. So I will tell you. Actually, the for this data we have cut down the data, right? So in real time, I will tell you. Just wait. Actually, I was working on the jagged one. So in this upregulated genes, you can see this jagged one. Can you see that? The gene
Yes.
name here. So it is showing you the log to fold value, this 4.3 something, right? And
Yeah.
also this JAG1, it is the upregulated gene here. So I was working on this particular one gene, that is JAG1 here. Okay. So we saw this is the upregulated gene. And also through KEGG pathway, can you see this figure?
Yeah.
So basically, it is involved in the pathway in the cancer where you can see it is having a connection with the delta notch. Can you see that
Yeah.
in this pathway,
Yeah. I'm
all of
seeing.
you?
Yeah.
So it is also have a link with the signaling pathway also. Okay, jagged one. Do you have any idea about the jagged one gene?
No, I don't know. I have never heard.
Uh, what about others?
No.
No, ma'am.
No, ma'am.
Okay, no problem. Actually, this jagged one is one of the protein, okay, that is interacting with the 4 receptor, and it is, you can see here, it is linking with the notch here. Can you see? So
Yeah.
this, it is triggering the notch signaling pathway, which is highly conserved pathway, okay, and further it can also lead to the cell dead also, okay. So on this paper, like we were working on the jagged one. So this is the main reason, like we are working on this KEGG pathway. Okay. So where we can see how the genes are interacting with each other. Okay.
Yeah.
Got it? Clear?
Perfect.
Okay.
Yeah.
So yeah, so from here also you can insert the picture from file. I don't know what is the name of that. Why I can't see. I don't know. Maybe you guys can later on try with this. Okay, you can insert it. I don't know why it is not showing. Okay, just leave it. We are not going to waste the time. Just practice it. Okay. Clear about this pathway enrichment, guys? Any doubt? Yeah.
Professor, just I have one thing about this. The
Uh-huh.
yeah,
Uh-huh.
the the images that about the pathway. Can we see?
This one?
No, no, the next.
This?
Yeah. Is there any way to change this image appearance?
Appearance? Not, sir. Not here.
It's like
You
that.
cannot. I don't think so. We can no, we
Okay.
can't modify this.
Mm-hmm.
We cannot. I don't
Okay.
think so. There is an option in that. You can see here, there is a help download
Okay.
pathway
Okay.
entry organism. We cannot.
Okay. Okay. Thank you.
Okay. Yeah. Okay, then. Next is the functional enrichment. So, any idea what is the functional enrichment now? Any one of you? Paul,
Yeah.
what is the functional enrichment?
I don't know. I don't want to say something. I don't know.
Okay. So, this functional enrichment is basically divided into three parts here. You can see biological, cellular, and molecular. Okay. So, now in through this functional enrichment, we want to see which function is highly enriched. Okay, and we are going to see the those gene which is involved and in that function. Okay, so for that what you have to do, mm, you have to load your NMIDs. Okay.
Wow. It is a big job. All this, it's not a small thing.
Mm, either you can do separately also, it's up to you if you want to create only for the down regulated genes, or if you want to create a separate function enrichment for the upregulated, so that time you have to put the NMIDs from the upregulated. Got it? So now
Yeah.
just load the NMIDs and then you need to select the organism. So it is belonging to human here. Just select the human. Then select the analysis. So I want in the form of pie chart. So I'm going to select this. Okay, pie chart. Then submit your data. Now, can you see this molecular function? Can you see this name?
Yeah.
Yeah.
And also, if you want to update to your cellular component, you can see the pie chart here for the cellular component. Then biological process also, if you want, you can see. So now you can see here in this biological process, can you tell me which function is highly enriched in this pie chart by looking this?
Cellular process.
Unclassified.
Which one, Hamid?
Unclassified. No pattern or category is assigned. The purple.
Okay, this one, purple.
Yeah. Yeah.
No,
Yeah. Yeah.
this one is
Okay.
not a sign.
Yeah.
I
Paul,
think
what
it is the
is the
blue one.
answer?
It is the cellular process.
Mhm.
Right, right. So this is the blue one, which is highly enriched in this biological process. You can see, Hame, there it is written unclassified.
Oh. Yeah.
Still, the category it is not assigned to those genes. Okay.
Mhm.
Now, if you want to retrieve more information, like if I select the biological regulation here, you can see along with the gene IDs. okay, which is coming from our data. You can see the gene names here, the panther families here, and this along with the species also. If you are
Yeah,
going to and the NMIDs also they are listing. Okay,
the same too. Yes.
then did you got this now? Can you tell
Yeah,
me for this, maybe for the molecular function now? Can tell me
catalytic
which?
activity.
Yes. What about others, Rakhi? Pragya, Lina, what about you guys? What is the answer for this molecular function? Which function is highly enriched here?
Unclassified.
Which one? Which which Pragya?
Purple.
This one? No.
Yes, ma'am.
See, Rakhi, what is the answer? Rakhi, are you there? Savanand sir, Akansha ma'am, what about you? Okay, no problem. So you can see wherever in this bracket there is unclassified, that means you'd have to ignore those, okay, category. So then next you can jump to this blue color. So this is the catalytic activity. You can see it has labeled with the blue color here also. And now if I want to save this data, what we have to do, you need to go to the export here. Then just create one folder with the name of functional enrichment. So this is the pie chart of your molecular function. So like this, you have to download for each function, biological process, just update the figure and then export. Okay. Now, any one of this you can open. Delimited. Next. Remove this, then finish. So this is only the percentage we are not interested with that. You can delete this. Now this is just a serial number. You can delete that also. And then up, you insert the row above and type here molecular function. And then number of genes involved. Now we are going to create a same pie chart here, okay, in this Excel sheet. So that I'm going to tell you how. Okay. Now again, this copy entire this A and B data. Then go to the data. There is option sort. Then custom sort. Then you can sort on the basis of number genes involved. And then make it Z2A. Okay. Now again, copy this. You can select this. And there is option insert. And can you see the pie chart here?
Yeah.
Yeah.
So make it to 3D, it's up to you. So same pie chart you can also generate through Excel. Can you see that?
Wow.
And also, if you want to see how many number of genes are involved here, that also you can display. So just select on this particular one, this figure, right click it, and there is an option add data label. So you can see. Are you getting this? Did you got
Yeah.
this? How to create? Or should I tell you again this part?
Oh, I got.
What about others?
Yes, ma'am.
Lina, Pragya,
Yes,
did you
ma'am.
got this number of
Yeah.
genes in? Okay.
Can I
Even if you
Yes,
want
ma'am.
to change the title, you can. What Paul?
things, for instance, for no panther category, there are 22 genes
Uh-huh.
on assign. Is it possible to completely delete to work just with genes that have been
Yeah, you can
classified?
delete this. Now, see, you can delete this from here.
Okay.
This 22 and number this pathway, you can delete this. Now, if, for example, delete this first. You can delete this. Then you can create the pie chart for this. Go to insert, pie chart, make it to 3D. Now see, and select on this pie chart, add data label. You will get that. Now see. Paul, are you getting this?
Yes, yes.
Clear? Okay. This is all about the functional enrichment, and there are also other database also you can try if you want. Go and check it. The Shiny G also. This is one of the database. The people are also using this database. So there are a lot of other also you can try and you can browse it for the functional enrichment as well. Okay. Clear? Any doubt till here?
Uh, Professor,
So, haha.
uh,
Yes,
just
Hamid.
just I have a question. How
Mm-hmm.
how uh, this result happened? I mean, we had 22 number of genes that was not classified. What is the
Mm-hmm.
reason? What is the reason that we got this?
Hamid, still the research is going on. Na? That is why they are not still like
Oh,
they have validated. Okay, still
okay.
they don't have any valid proof or evidence,
Mhm.
you can say.
Okay.
Sometime in the NCBI also, if you see, for example, I want to work on this BRCA2 gene.
Okay.
Actually, this BRCA2 it is causing cancer in ovary. So see here, where it is? Yeah. Can you see this ref sex status?
Yeah.
Can you see the reviewed word here? So can you tell me what is the difference between reviewed and unreviewed? Can you tell me that?
Yeah, the unreviewed is on the processing that it's not complete.
This one, reviewed?
No, no, that's unreviewed.
Yes, sir. So
Yeah.
reviewed means there are like lot of people, or you can say the expert panel who is handling this NCBI. Okay, so they are from like the from the wet lab till the last analysis they are doing, or they are working on this particular one gene. Okay, so
Okay.
after like having some wet lab proof, also valid proof, you can say all the validations have been done for this BRCA2. That time only they are assigning this as the reviewed, sir. Okay,
Yeah, yeah.
so that is the reason, like if you are asking for this pathway also, the one you have seen here in the unclassified, that means still the research is going on for those particular gene. It can happen, right?
Okay, okay, okay. Thank you, thank you.
Yeah. Or you can say the unclassified are genes are those are just a predicted one. Okay, just assumption, we can say. That's it, assumption for them. Clear?
Yeah, clear. Thank you.
Okay. Next part is we are going to see the network analysis tomorrow. But before that, everyone is having this cytoscape tool. This one, this app, are you guys are having?
No.
No?
No, ma'am.
Okay. Okay, then what you guys can do, just go to the browser here. So make sure everyone is having this cytoscape tool tomorrow, because you are going to perform along with me.
Professor, performing along with without you is worse, I think.
No problem, I will go slow, sir.
Okay,
Okay.
okay,
Yeah. Actually, we are going to create a network tomorrow. Okay. So that is why I wanted you can do along with me. Otherwise, it will create a problem afterwards, because we are going to create one new Excel sheet also. Then only we are going to load here in the cytoscape tool. So why we are using this cytoscape and the string database, I will tell you the difference maybe tomorrow. Okay. So till the
okay,
time, what you guys can do, just right here cytoscape download and go to this older version here, and you can see here 3.8.2 version. You can select this.
okay.
Now whoever is having the Windows system, so I know like already like the people who are using the Window, they are must having the 64-bit processor in them. So you can go with this, just install this in your system. And the one who is having the Linux platform, they can go with this. And for the Mac one, they can go with this DMG file.
Can you
And
repeat, please?
yeah, yeah. So Paul, you guys are having the Windows system. I know maximum users are the Windows one. You can go with this link and install that tool. This one,
Okay.
this one. Even if you want to check with either it is your 32 or 64 bit processor, you can just check it from the settings also. And just type here about. Can you see this?
What?
System type 64 base processor.
Yeah.
So this is how we are checking the configuration. Got it? So
Yeah.
please download this and make sure you guys are having this gene expression file. Okay. By tomorrow, you can please practice till this functional enrichment. I will check it. Okay. Tomorrow.
Okay.
Yeah.
Yes,
So
ma'am.
it's a homework. So everyone, make sure everyone is having this Excel sheet, and along with the you need to sort this upregulated and downregulated everything. Okay. Till this step, you have to perform the pathway enrichment. Okay. I will just check it tomorrow,
Okay.
and tomorrow we will see the network. Okay.
Ah, sorry to interrupt. Like that Excel sheet, we need to generate by running these commands, or like you have shared via.
Sir, I have shared this also. Just wait, I will tell you. If somebody who didn't executed this pipeline till now, so
Ma'am, can I show my screen?
mhm, just wait, Pragya. So in this module 7, like I think all of you they are having the access of this reference base, right?
Yeah.
So can you see, sir, Sarvanand sir, this file gene expression diff file?
Yes, I'm having, yeah.
Yeah, so this is in the text format. You can open this in the Excel and you can perform all this, okay, in your system also.
Okay, got it. Yeah.
Okay, okay. So make sure everyone is having this cytoscape tool also. I will guide you tomorrow for the network analysis.
Okay. Thank you.
Okay then.